a whole set in these last bit where there's just little questions in there, and so there's like some questions that kind of look like essay questions, and some of them that look like multiple choice questions as we finish off this lecture. Just a heads up, we do have that bioinformatics exercise in there.
I couldn't get that to open.
You can get it to open.
Yeah.
Oh gosh, I was gonna ask you guys. So I'll probably extend the due date on that one for sure. Let me see what the deal is really quick. I better figure out the figure out what's going on with that before I forget. So I need to re-upload. I have the PDF version, so that's the problem, I think. I knew, we talked about that already, didn't we?
Yeah. And I probably see the point.
20 minutes? Oh my gosh, that is okay. Okay.
Last time. I think, well, we do have a quiz
Alright, I just put a note for myself. I don't want to take your time to fix it right now. But I will fix that, and then what did I have as the original due date?
Monday or Sunday.
Sunday? Okay, we're going to push that into Wednesday of next week so that you can have some time to talk with each other. Alright, anyway, so we'll either, since we're going to push it off anyways, we'll either, until you can have a chance to look at it, let's get together Friday to pick our groups. Or you can be talking to each other now. We'll do our groups again. Okay, any questions about transcription initiation? So there, I sat with somebody today. It is a transcription initiation. There's a lot of stuff going on. So it's not a bad idea, just a little exercise that you can do to start kind of making it feel a little bit more comfortable is to get in there and, group together the events. So what are the proteins that bind, here's just a couple of questions, just jot them down and see if you can build a flashcard or something to help you with. So, what are the proteins that bind the small subunit of the ribosome for initiation?
Which one, prokaryote or eukaryote?
Oh, the question for prokaryote or eukaryote. You want to know for both of them. Yeah. And then also, there's like one protein that's important for binding the tRNA, and it's a GTP. Yeah. EIF2. It's a GTP bound protein. Okay, and then in the eukaryote, the extra thing is that complex that's holding the mRNA. So can you answer the questions? What are the proteins in the complex for RNA? There's a number, like one number, and then a bunch of the letters that come after. But it's EIF4, right? So we've got 1 and 3 on the small subunit. We've got 2 on the tRNA. We've got EIF4s on the mRNA. Okay, so that's kind of how you got to group it. You got to put it together a little bit. If I'm going to be honest with you, if you get an essay question where you got to describe this stuff and you get the letters mixed up, I'm probably not going to take any points off for that. I'm not trying to make it like say I'm going to let you just learn the wrong thing, but just so you know what the level of fear is there. Multiple choice questions, they, with all of these little extra things, they might jog your memory actually and help with that stuff. Okay. Any questions about elongation before we talk about termination? So remember the A, the P, and the E sites, and then we added in a couple of other proteins in that process. So we've got our EIF2 always, right? And then we've got the GTP switcher, right? So we've got to take the GTP, the used GDP off and put the new GTP on to turn that protein on and off. So we can grab the new tRNA from it, right? Amino acids can be like thousands of them come together in a chain. So you imagine there's like 1,000 tRNAs that have to be brought in. So how many can one EIF2 do? You know, probably around that, 1,000. That's good. Okay, so then now in termination, we saw the code we saw the coding sequences in that table, and we know that there are some 3-letter codes that are dedicated to they're the stop codon, or sometimes we call them the nonsense codons. So there's no tRNA, there's no amino acyl tRNA that's going to be loaded with an amino acid that has that anticodon. So there's none of them that will bind that. Okay, so that elongation is going to continue till it reaches those, and I'm just going to rattle them off. Of course, you'd have a little table to help you, but it's the UAA, the UAG, and the UGA are those, and they're in the notes if you're looking at those. In eukaryotes, we have something called a release factor that will bind those sequences. So prokaryotes and eukaryotes have these release factors. So this is that little that purple protein that you're seeing in here, and there's something that happens when they bind. So here we are, we've translocated to a UAA, which is a stop codon. There's no tRNA that binds, but instead a release factor is able to bind the A site at that moment. And upon binding, there is a couple of reactions that happen. So when this thing binds, instead of building a new peptide, there is a chemical reaction right here that releases the peptide. There's no new amino acid to join to, so the chemical reaction happens basically with water, and then it goes off of the thing. So once we release the peptide, then the next thing that happens is everything else that's bound together falls apart. So our mRNA lets go, our large and our small subunits let go, and our tRNAs are expelled or released. Okay, and then we can do it all over again. If these aren't damaged, right? So the mRNA probably will get chewed up and removed, but all of these things can go to the beginning and start all over again. Okay. Of course, this thing would have to bind what? EIF1. Yep.
1 and 3.
And then all that stuff would happen again. Okay, so we really looked at it simply like one ribosome for one mRNA, and that's not really how it is. You can have many ribosomes marching along along your mRNA. So we have polyribosomal complexes. So here you can see like we've got one. These are actually 4 ribosomes on the mRNA that are marching on. This is actually an electron microscopy image showing that structure, the polyribosome complex. So each of these big like snowball things is an mRNA, or is a elongation ribosome marching along. Right? So each one of them came in and initiated in the same way, and then when the first one got out of the way, the next one came in and initiated at the 5' cap, and so on and so on. Okay, so every level of gene expression is regulated. You know, we've already talked about regulating transcription, and now we're in translation. So this is a structure that can be formed. So remember that initiation complex had this kind of, the mRNA did the same thing. There was like, instead we had the poly-A binding protein here, we still had the EIF4E binding the 7-methylguanosine cap, but then we had some other 4s in here that were binding, B, G, and I think were binding in there, right? There's another protein, if it's available at a given time, that can take over the space, and that's just a transcriptional repressor. Okay, so this is a key thing for regulating gene expression at the translational level. This is a global translational repression. What do we mean by global translation? It means that this can bind any of the mRNAs. It's not sequence specific. It's a way to shut down translation. I have an interesting story to tell about some of the research we do in the senior level cell biology class. I had a group one year use a drug they called it juglone, was like the common word for fishermen, like they use something called juglone. It's actually a quinone. It was really interesting because it was a regulator of gene expression, and that was literally what we saw. Like we treated the cells and gene expression went away. So we had like this cool RNA experiment and stuff where like looking for things, and we had nothing because it shut things down. So it's basically something that bound things. So that it was doing something similar to this, right? So and it was global because it shut down all gene expression, which is probably the reason why. So that compound is in walnut, the husks of walnuts, and if you ever walk around and see those walnut trees growing, when those husks fall down and then degrade in the ground, you'll notice nothing grows under a walnut tree. Probably because there's no gene expression. That's possible. So it's it's global. It's all the gene expression. Yeah. So okay. All right. We already learned about this, but this is just another reminder that there are sequence-specific ways to target translation. And so mRNA is a target of translation. We talked about it in RNA processing and all of that stuff because it is an RNA processing mechanism. But remember that it's not preventing RNA transcription, it's preventing RNA translation. And it is specific because the way that it works is this RISC protein has to be loaded with a specific sequence. And then that specific sequence finds the mRNA that it is targeted to, and it degrades just that one. Okay. So there was that cool, I think I shared this story with you before, there was that cool story where there was a group of gap junction researchers that used this same mechanism to specifically shut down the translation of connexin 43, a gap junction protein. And they did this for patients that were receiving corneal eye transplants. And just shutting down that one protein because of its ability to help with migration and targeting white blood cells and things like that, that allowed them to have more success in getting those transplanted tissues to take. So, and what would happen if they used a global thing? They would definitely not work. But they were able to target a specific protein with or a specific gene expression with microRNAs. Okay, so now we're putting in our microRNA. We're adding to our microRNA story what we have now learned about the way that the ribosome is handled. Okay. So just to give us a better picture of the molecular status of this can still happen. You can still have initiation. Typically, the microRNAs are targeted to somewhere along the mRNA. Okay, so that will be degrading that, but it doesn't directly inhibit. It doesn't directly inhibit the binding of the initiation complex, but it does inhibit translation because even if this happens, if there's no sequence to read once it reaches this position, then there's no translation, right? We can make small pieces of proteins and they can get degraded in the trash can later. But if this is active and it's a race, once the RISC complex starts to chew everything up, then it's gone. There's nothing to initiate on. Okay, there's another global repression mechanism, and one was targeting, the first one we were targeting the mRNA specifically, right? And the initiation complex proteins that are bound to the mRNA. The other thing that can be targeted to globally repress translation, I gotta remember these words, translation, is by targeting our EIF2. So our EIF2, remember, is the initiation factor that's binding the tRNA and bringing our tRNA in. The first one is always bound to the methionine tRNA, aminoacyl tRNA. And so something that can be, remember that the way that this works is we've got EIF2 on, we have EIF2 off when it's GDP bound. And the only way to turn it on is to have the switcher. So this might be a little confusing because in our last slide we had a little word that said EIF2 beta gamma. That's the same thing. Okay, so just if you want to shorten it to B, EIF2B, this is the switcher that takes off the GDP and gives us the GDP to get us back to the beginning of this cycle. Okay, so what are we saying? We're talking about global inhibition of translation. So what do we do? We can stop this from getting turned back on by not switching out the GDP for GTP. Does that make sense? So wordy, I know. So 2 ways to globally stop translation. Don't let the mRNA bind the initiation complex by messing up the EIF4 association, or don't let our EIF2 bring in our tRNA by preventing it, by not turning the on switch on. Not letting it have a GTP. Okay, so this begins the next part of this lecture. We're going to go through some questions, and hopefully I can get some of you guys to answer them. I know we're all really tired. I dropped my little nephew off at school today, and he is the most joyful little person that I've ever known in my life. He is like, I dropped one kid off to my parents, and then he's always there, and he's having breakfast and stuff. And if I have the time, I'm not rushing. I'm like, come on, James, let's go. I'll take you to school. And he's always like, so many things to talk about, but today he was just like, And usually he's like, just bounces out of the car. He's like, I see my friend, I see that friend, I'm gonna go join. And he was just like, I don't want to be here. So it's like a normal feeling. It's spring. Everybody's got spring fever. But anyways, hopefully I can get some interaction as we go through here. But before getting into, so we're going to look at, reminding myself, protein folding and regulation. And before we even think about it, we got to remember some of the things that we've talked about before. So taking it all the way back to the beginning, remember we introduced cystic fibrosis. Like, what is the deal with that? That's where we have, we have transcription and translation of a polypeptide. We get to that polypeptide stage. That string has to be folded in the correct shape for it to have for it to go where it needs to go and for it to do the job that it has, right? So the cystic fibrosis receptor is one that is well known to be mutated so that it doesn't get that correct shape. There's a misfold in the protein. And the problem with that is it's actually involved in regulating the salt concentration inside and outside of the cell because it's involved with it's involved in ion transport and at the membrane. Okay? So remembering how biological membranes work, nothing charged can pass through a membrane. So if we don't have that protein there, then we don't have the right concentration of salts in the area. We get cystic fibrosis patients end up with like very sticky mucus because the water isn't being regulated. The osmotic pressure isn't getting put there like it's supposed to, or it's being overwhelmed. So there, now that we're thinking about gene expression, we want to remember some of the stuff. I feel like I'll be out of order here, but then we're going to go into these things a little bit more. We've already talked about some of them, but remember the nucleus and how what is the structure of a nucleus? We have that double membrane structure. we've got to get our RNA out of the nucleus. And then now where, we just talked about translation, all the molecules involved, but where does translation happen? Especially if it's a membrane-bound protein or if it's a cytosolic protein. What is the difference? Okay? So the ER and the Golgi are going to be really important, especially for cystic fibrosis, because it is a membrane-bound protein, and it has to be built in the membrane. So we're going to look at that mechanism a little bit. And then the cytoskeleton is important because we have to traffic things, and sometimes the cytoskeleton is helpful with that and give us the shape to the cell. Okay? And then don't forget that all that we did cover a lot of the structures in the cell structure class, in class exercise. So let me know if you have questions about that. I know that it was kind of scattered because it was like a lot of groups teaching us. So anything that just needs a reminder, don't hesitate to ask. Okay. Okay, so protein folding and regulation. This is a really cool model of a protein. Actual, like, you could almost get even more detailed with this and actually name the amino acids, and then you can follow them. But this model is giving us that three-dimensional, how those amino acids are taking up space in a three-dimensional space, right? So in this part of the lecture, these are the things we're covering. So protein folding and processing, and then regulating protein function by changing the shape, and then protein degradation. How do we get rid of those proteins when they're done? How do we break this thing up into amino acids? Or just carbons, or anything like that. Okay, so we saw the polyribosome complex, or we saw the ribosome at work, and we were, in our last picture, we saw the blue polypeptide coming off of there. But the polypeptide isn't the three-dimensional shape, right? We need something that has a specific shape. So the new molecule we're introducing right now is the chaperone protein. And the chaperones, these are really neat because they bind very specific sequences along the polypeptide chain, and it basically prevents them from coming together prematurely. So say you're just to give us a more molecular picture of this, so say we've got, a hydrophobic region here, and we have another hydrophobic region there, but these two aren't meant to come together because they're different domains, because they're going to be on different sites in the end once the proteins are made. So these chaperones will come in and they will bind in a way that prevent those two things that want to come together to come together. Okay? We have to protect against abnormal folding from this stage so that we don't get that misfolded protein and, we have a major malfunction in the cell. All right, so once the chain is fully made, then those things can be released, and that is also even a stepwise manner to do that. Okay, so the chaperones are the ones that actually bind the peptide, and then then how do we remove the chaperones and fold it properly? So here is our mRNA and our polypeptide coming out. We have the chaperones adding to the polypeptide. We've got there can be some partial folding. Notice that not every single amino acid is bound by a chaperone, but there are, you can get some alpha helical structure or some of that structure that comes right away. But the overall total folding is going to happen in this cool giant complex called the chaperonin. So this is the greatest thing about biology, right? All these words that sound exactly the same. But the chaperone, just keep that right in your mind, is the thing that binds the peptide. The chaperonin is the large complex that's going to be involved in helping that final folding conformation. Okay, so the chaperonin is basically there are a number of subunits, and they're arranged in these stacked rings. They're that's what you're seeing here, these stacked rings of protein subunits. The transfer to the chaperonin is ATP dependent. So moving this thing in here and putting it into the chaperonin is ATP dependent. Whenever you're seeing ATP at this level, we talked about this in other classes, but maybe it's not put into this perspective enough. Anytime you're seeing an ATP that is being hydrolyzed and into ADP, we're doing some sort of work. And you got to take yourself all the way back to that second law of thermodynamics. You have to have in your head is anytime we're taking things that are nice and simplified and spread out, taking up space, and then forcing them into a shape where they don't get to take up a lot of space, we have to put energy in to do that, because that is energetically unfavorable. That is a going from stable to an unstable state. Isn't that weird? Proteins are unstable, but that's what it takes. And taking us back to metabolism, this is anabolism, right? Okay, so you're bringing this piece to this piece, we're bringing these things together so it's ATP dependent, and then it is ATP dependent within to actually get all the correct amino acids to link up with the correct amino acids in there. And then you get your properly folded protein. And a lot of that is, so this is an assister, these are assisting as well, but don't forget that the overall shape of the protein is still sequence dependent. What do I mean by that? Those amino acid side chains, the acidic ones, the basic ones, the hydrophobic, hydrophilic ones, the way that they are situated is going to drive that overall shape to the protein. All right. So local folding produces secondary structure. I mentioned them already, but what are the two major secondary structures that we remember? This is just a review from way back when. What are the two structures? Secondary structure. I think I heard them. So one of them looks like this, and one of them looks like a pleat. Do you remember those? I love how that's moving. Yeah, alpha helical beta sheet. Yeah. So this question helps just looking at this picture. That type of folding is local folding. So that means like these amino acids that are right next to each other, that sequence is going to drive that. Because what is that, just taking us back, what is that dependent on? It's dependent on the covalent bond, the N to C terminus bond, and how that thing is angled. Okay. All right. So just a reminder, just because we're thinking about protein folding, what does that secondary structure look like? I don't have a picture, but go back. Is it chapter 2? Remember that it's hydrogen bonding that holds some of that together, some intramolecular hydrogen bonding. Is it the side chains? Are the side chains involved in secondary structure? No, that's correct. I see some no's. No, the side chains drive that tertiary structure. Okay, so there are a lot of diseases that are associated with misfolded proteins. Literally, the disease status is because some protein didn't get folded correctly. So there's cystic fibrosis, Alzheimer's disease. So cystic fibrosis remember it's the cystic fibrosis, the CFTR receptor, the RS4 receptor. The misfold happens within the ER. The most common mutation is that delta F508. There's a phenylalanine in the amino acid sequence at position 508 that's missing. And that is so without that, then in the chaperonin or whatever helps to fold it, it's not going to fold properly. So the sequence is missing. Alzheimer's is a disease where we have misfolded amyloid protein. So here's a picture of the native state of the amyloid protein. So notice all the alpha helical structure. There's a problem with the sequence where we end up, it's either the problem with the sequence, or there's a problem with the way that the chaperones are binding, or the way that there's just a problem in the way that the thing comes together in its shape, but it causes these beta sheet structures instead. And this causes brain damage because we have a bunch of these that are don't get degraded and they develop plaques. They fill up the cell dies, basically. So that's a problem. So that's Alzheimer's, which is the amyloid protein. It's a specific protein. Prions are kind of more, spread out. A prion is what it is it is a misfolded protein, and the difference between this one and Alzheimer's is the scariest thing ever is that this thing can self-replicate. And it's weird. So when we talk about self-replication for this, I don't know, I feel like it can be described a little bit better. The way this works is you have your native protein that is expressed. You get transcription, translation, and you may get the native protein. But once you have this prion, it can actually bind other native proteins and turn them into the same shape. Does that make sense? So the misfolded protein binds the other proteins and it changes the shape so they look the same. And they bundle up. So here we're actually showing amyloid, so like the Alzheimer's one, but there are many other proteins that have been identified as prions. So the diseases that have been linked to this scenario are scrapie, mad cow disease, and Creutzfeldt-Jakob's disease, and Kuru. Kuru is one I have that's kind of newer to me, but. So protein folding is important, right? And the way that it's regulated is important. Okay, when we're processing protein, so just like we processed mRNA, you get a sequence, you get it, and you need to, when we talk about processing, we have to do things like change the sequence or change the shape. Proteolysis or proteolysis is where we actually will cut the polypeptide chain. That's what it is. An interesting thing about protein processing is that sometimes we will actually, as the ribosome is kicking out a polypeptide chain, sometimes there are pieces that have to be cut off, and that is even before folding. So proteolysis is actually a mechanism or a part of the mechanism for getting that properly folded protein. So some proteins are going to have what's called an amino terminal signal sequence. What do we mean by that? Remember they're N terminus to C terminus. So on the N side, there is a signal sequence, a known sequence of amino acids, and there will be an enzyme that will cut there. Okay, so let's walk through the picture because I think that makes it a little bit easier. So here we have our mRNA being translated, and we have a signal sequence coming out. Okay, so it's their peptides are built N terminus to C terminus. So we've got the N terminus coming out, and this is a membrane or protein that's got to get through a membrane. Here's our ER. This is our first picture of the location of this stuff. By the way, we'll have a couple of them. But here we have a protein that a transporter in the membrane of the ER that's responsible for getting this protein in the right place. The N terminus, as it's transcribed, is being pushed into this channel, and once it, notice it's not like coming in there perfectly where it's like this, the very first one is going first. It's actually held within the transporter. The transporter itself is going to position this properly. So it's held there at that end, and as it's being translated, it's being fed through. And once we get to the right length, that signal sequence is recognized by our peptidase. So peptidase, ACE meaning it's an enzyme. A lot of times it's a cutting enzyme. And what is it cutting? A peptide. So we get help from the word here. We actually get help from a biological word. So peptidase. All right, so this is where the cut happens, and then this thing can be degraded or removed at some point, but here is the actual first part of the protein that ends up in there. So where was our F-methionine? It's down there. So we might, in the end, we might not have that at the beginning of our protein. Okay, so that gets continuously transported across that membrane so it can be in the right place. Okay, so question number 2. Some proteins are translated directly into the ER. What happens to the protein in the ER? We talked about folding, so just, we'll get more into detail with this, but just a picture to keep building in your mind. We talked about chaperones, and chaperones are going to be in the lumen of the ER. So as that was what was missing in this picture, is you have chaperones that can link on as that's coming through, and we can have proper folding in the ER. So this is a protein. Another thing to just point out, this as we're thinking about this is a protein that is not embedded in the membrane. It's not this one that we're looking at right now is not a transmembrane protein. Okay, we don't know for sure if it's a peripheral protein. We don't have anything to tell us, but it is a free-floating cytosolic or a free-floating secreted protein is what it might be. All right. So we have a picture now that, translation happens on the ribosome. If it's something that it has to go into the endoplasmic reticulum, we now have a picture of how that translation and that protein localization are connected. All right. All right, so we're processing proteins. This is a picture of insulin. So insulin is interesting because this is just an example, but there are many proteins that might be formed with like these long. They're just kind of like the RNA. I'm jumbling my words. I apologize, but think back to RNA or eukaryotic RNA. Sometimes we take some of the sequence out, right? So there's a similar thing that happens with proteins. Sometimes, we get the full polypeptide chain, and another thing that happens, we aren't just cutting off signal sequence for proteolysis. Proteolysis is also part of processing when we need to remove some of the sequence too. And that because the final shape doesn't have that sequence in it. So here is insulin, and so we have the nice diagram of the peptide from N terminus to C terminus, and we have a signal sequence that we already saw in the last example, but then we have this like B portion and this A portion, that these are the green is the actual final protein. The red we cut out. And this might be something that's evolved to change the shape of the protein. It might be, I can't tell you the evolution reason exactly, but in the end, these are the pieces that we need. And we need them to come together like this. So notice how when we have proinsulin, we have the full sequence they're all linked. So our B region connects end to end to the A region like this, but then there are these disulfide bridges in between those two parts. And then we can have removal of the connecting polypeptide by some lysis, protein lysis, some sort of peptidase enzyme to cut that. So proteolysis has its role in processing our protein. Okay, there are other things that we can do to change the shape of the protein. So the sequence is important. I know we've hammered that in, but we also can change the shape of the protein by adding other organic structures to the protein. So glycosylation is where we're going to put on sugars or carbohydrates. And when we have a peptide with a carbohydrate added, that thing is a glycoprotein. Since we're talking about adding sugars to anything, it's worth mentioning that there are oh my gosh, I am like going all over the place. Don't look at that yet. The same way that sugars can be added to proteins, they can be added to lipids too. So then you get a glycolipid in that case. So you get glycoproteins and glycolipids. Okay, lipoproteins are when, we can add sugars to proteins. We can also add lipids, fatty acids, to proteins. And so then that would be a lipoprotein. Okay? So here's glycolipids. So for glycolipids, these sugars are lipids linked to oligosaccharides. Glycolipids, those things that are the complex thing that is, that can also be added to proteins. So you can have like both things added to the protein. These are typically added to the C-terminal domain or tail of proteins. So glycolipids can be added to the C-terminal tail of proteins. So these are complex structures, and these are things that we can add to the protein to change its shape and then change what it's able to do. If you add a lipid to a protein, what can it do? If it's not a transmembrane protein, it can embed it can embed the lipid in the membrane. It can be linked up to the membrane, right? If you add a sugar to a protein, you know, it can change the signature of the cell by being at the membrane, and you know, you've got a specific sugar that your white blood cell is going to recognize as something. Okay. So here's just a picture of that stuff. So here we have our glycoprotein. It's being translated. There is a specific amino acid right here on the near the N-terminus where we have the addition of these sugars. This kind of stuff happens. There's a core sugar that's added within the endoplasmic reticulum, and then we can change this, add more and more of these little monomers of sugar to change that overall structure in the Golgi. Okay. Here's a lipoprotein that's myristoylated. So here is our amino acid, our peptide, and our amino acid sequence. So this is how it's built. You have removal of the initiating methionine. So here we removed our methionine here. We have myristoylation of the N-terminal glycine. So we've got a glycine, and there's a chemical reaction to add this fatty acid chain on there. So RAS proteins, if you're like interested in cell signaling at all, RAS proteins that get myristoylated, and that really drives that type of signaling in the cell because that protein needs to be at the membrane at a certain time. And then to turn off the signaling, you cut off the fatty acid chain, and then it's not in the right place. Okay, so what is this one? I'm skipping over it. Oh, this is a lipid. Here is a glyco. This is the glycosyl phosphatidyl inositol, the GPI anchor. So here is a complex lipid. It's a membrane lipid. We've got our phosphate head group, our two fatty acid chains. Attached to the phosphate, remember you can decorate the phosphate further, we have a glucosamine, so some sugars. And then we have an ethanolamine, an organic structure, and then we have this linked to a protein. So that is a very complicated structure right there. It's got all of the things, all the macromolecules, proteins, sugars, and lipids in there. So that's a GPI. This is a GPI anchor. All right, so it is 5 minutes. Regulation of protein function. Okay. We've already talked about how we can stop translation, we can stop transcription, and all that stuff. So we can regulate the amount of protein in the cell at that level. But you can regulate the amount of protein too on the back end of it, like sending things for degradation. The other thing that we've already talked about is turning things on and off with that GTP, right? So that is a pretty standard mechanism to regulate the activity of the protein. So these are the three general mechanisms. You can regulate our proteins by small molecules that might be binding them. You can phosphorylate, or add a lipid or something, like you can change the shape with these extra pieces that you can put on. And then they can be regulated by protein-protein interactions. So like sometimes we have proteins that are constitutively expressed. They're in the cell, but they're bound to another protein and they're off. And only when you release that protein-protein interaction do we have the on state. So that's not a GTP thing, that's actually another protein binding. So typically the mechanism for turning something on is is a feedback inhibition mechanism. So feedback inhibition refers to, we know in metabolism there's all these enzymes doing these different chemical reactions, and the chemical reactions are linked together. In feedback inhibition, you may have a set of reactions linked in that way, and in the end product has the ability to go to the first chemical reaction in the pathway and turn it on or turn it off. So that's what they mean by feedback inhibition. Something down the line goes to the beginning and regulates it. Okay. Feedback inhibition is often allosteric. What does allosteric mean? This is I'm pulling things that come from earlier classes, so it might be a little tricky, but remember an enzyme has an active site. It might have a site outside of the active site that's bound by something. So oftentimes feedback inhibition is that type. There's an allosteric site that's bound. All right. So let's go ahead. I feel like we need to stop here. What do I have left? I have like a few slides left that we'll get to on Friday.